Hi, today's lecture is going to be about the scientific method. And this is a process that is taught in most biology classes or most science classes. And it is under the assumption that when you do science, you go through these steps, but rarely is that the case. Real science is messy, and usually one thing leads to another thing, leads to another thing, or something gives you an idea out of nowhere. And then you find your discovery. And so rarely do you go through the steps in a really clean way, like I'm about to describe. But the purpose for going through the steps of the scientific method is because once you make a discovery in science, you want to publish it to share it with everybody else. And when you publish a paper, you write them in these steps. So whenever you read a publication of somebody's discovery, they are written in this order. So for that reason, it is important to learn the scientific method, which is the title of our notes. Okay, and depending on which class you're in or what textbook you're using, determines how many steps that the scientific method is described as. I'm going to describe it in 6 steps because that's what I like. And so the first step is to come up with a question. So you're going to ask a question. So a lot of times you'll be going about, life, and you'll make some observations, and something will spark your interest and you'll start wondering. And this could lead to a question. And so a question is something that has a question mark. And I know that's silly to say that, but sometimes I will ask students for a question and they write a statement. So a question has a question mark. Okay. And I am going to use an example throughout this lecture to just illustrate each step. So I have a tomato garden, and I'm going to show you a picture of my tomato garden. Okay, so I'm going to show you a picture of my tomato garden, and let's say that I was hanging out in the backyard and looking at my tomato garden, and I noticed that one tomato plant made so many more tomatoes than the rest of the tomato plants. So here is the tomato garden. And let's say one plant produced a lot more tomatoes than the rest of the plants. Let's say the rest of the plants made like 2 or 3 and one made like 20. And I started to wonder, I wonder why that one tomato plant made so many tomatoes. So that would be a question that I could actually answer using the scientific method. So my example will be, why does one tomato plant make more tomatoes than the other plants? Let me scoot my picture over a little bit here. And so my question, it has to have a question mark. And there's my question. Okay, I'm going to use that for this topic. So then I'm going to move on to step number 2, which is do your research. Okay, so you're going to go onto your computer, or if you're old fashioned, you're going to go to the library and look stuff up in books. But what you're going to do is you're going to start researching everything there is to know about plants and tomato plants specifically. You are going to definitely look up, has somebody asked this question already, and is there already an answer? And if there's already an answer, then you just look up the answer. But if there isn't an answer, then you should go through and do the experiment and maybe find something new that you could share with the world. So I'm going to do some research, and I'm going to look up. So for my example, okay, so look up information about plants in general and tomato plants. Okay, and I'm going to look up, what do plants need? So I'm going to find out that plants need sunlight, and they need water, and they need minerals in the soil. And I'm also going to learn some things about pruning. Pruning might make a difference. So based on my research, I decide I'm going to try and see if pruning is the reason why that tomato plant made more tomatoes. So then we move, after doing all your research, you move on to step number 3, which is form a hypothesis. And a hypothesis is a statement that is a potential answer to your question. Okay, so it is almost like a prediction. You have this question, which is, why does one tomato plant make more tomatoes than the other plants? And then my hypothesis is going to be a potential answer to that question. And this is all based on the research that I did. So for my example, oops. So for the example that I'm using, I'm going to say pruning. So if a plant is pruned, then it will make more tomatoes. Okay, so there's my hypothesis. If a plant is pruned correctly, then it will make more tomatoes. And that is what I'm interested in looking at. Okay, so now I've got my hypothesis and I need to move on to step 4. Step 4 is design an experiment. And when you design an experiment, you need to keep in mind what the variables are going to be. So you need to have an independent variable. And the independent variable is the variable that will be different between the groups. So the factor that will be different between the groups. So in my experiment, I'm going to have a group of plants that will be pruned and a group of plants that will not be pruned. And so my independent variable for my example is pruning or not pruning. Okay. And so let's say I'm going to design this experiment. I'm going to have 10 plants in this group and 10 plants in this group. And the plants in this group are going to be pruned and the plants in this group are not going to be pruned. But all the other factors are going to be exactly the same. So they're all going to get the same amount of water. They're all going to get the same sunlight. They're all going to get the same soil, same environment. But the only factor that will be different is whether they're pruned or not. And so that means that is my independent variable. And then the other variable is called the dependent variable. And the dependent variable is your outcome of your experiment that you are measuring. The outcome of your experiment that is being measured. And so this also has to be decided before you do your experiment. And for me with the tomato plants, the thing that I'm going to be measuring at the end is the number of tomatoes. Okay. And so there are lots of things that I could measure. I could measure, how tall the plants are, or I could measure how red the tomatoes are or how big the tomatoes get. But you have to determine what your dependent variable or variables will be before you do the experiment. And for my experiment in this example, I'm deciding that I am going to do the number of tomatoes. Okay. And so another thing is when I do my experiment, when I do this experiment, I have to make sure that there's no like bugs that get in my garden, because I might have a praying mantis come and eat my tomatoes, and that would be bad. And then I would have to redo the whole experiment again. And then that would just be so hard. Oh my goodness. So you got to keep all the bugs out of your tomato garden. Okay, so moving on to the next step. So once you design your experiment, guess what the next step is? It is do the experiment. So step 5 is do your experiment and collect your data. Okay, so that's what you're doing. You're trying to get data. You cannot do any science without data. Okay, so that's like the most important part of science is getting the data. And when you are collecting data, there are 2 types of data that you can be collecting. Okay, 2 types. The first type is called quantitative data. Quantitative data is any kind of data that has a number. So it's called numerical data. Okay, numbers. If you have numbers in your data, that is called quantitative data. I am collecting in this experiment the number of tomatoes, and that is a quantitative data. So if you have numerical data, you use statistics, to make sense of the meaning. Okay, so you have to use statistics to see if it's meaningful or not. Also, you graph numerical data on a line graph. Okay. So whenever you collect data, you put it on a graph, because just looking at a bunch of numbers doesn't mean anything. It's hard, or it's hard to see the meaning. So you put the numbers on a graph, and now you've got a picture that will highlight any kind of pattern that might be emerging from your data. The second type of data is called qualitative data. Qualitative data is non-numerical, and it's also known as categorical data. And these are things that are not numerical. So, for example, if I were to say the redness of a tomato, so the color of the tomato, or the texture of the tomato, like is it crispy and crunchy, or is it mushy? Things like that. So anything that is not numerical. And when you have qualitative data, you graph this data on a bar graph. Okay. Okay, so now you have your data, and then you put it on a graph. And when you see the graph, that means you're analyzing your data. And so you analyze the data by doing statistics and putting it on a graph. And from that analysis, so from the data analysis, you form a conclusion. Okay. And that's the last step. You're forming a conclusion. And from this analysis, you only have two options for your conclusion. So two options. You have first option, which is reject the hypothesis. So what that means is you have this hypothesis that pruning means more tomatoes than non-pruning. And if that's not what you get, then you reject the hypothesis. You say, I do not have data to support this. And the second option that you can do is you can fail to reject the hypothesis. Okay, and this means that you do have supporting evidence. Okay, so this means there is supporting evidence. And so you would never say, I have just proven that pruning tomato plants means more tomato, because you can never prove anything for certain in science. Okay, so you just say, I have supporting evidence to suggest that pruning means more tomatoes. And so I fail to reject my hypothesis. Okay. And so that's it for today. I hope everyone's having a great day, and I will see you next time.